My name is Josip and today I'm going to talk about clinical shotgun proteomics in cancer biomarker research and drug targets research. I will be talking first about cancer biomarker and drug target discovery in general. Using MS-based clinical proteomics will be following. I'm going to discuss challenges, limitations, solution advances that we are trying to apply in our research, the application, and conclusions. So nowadays, majority of cancer treatments, unfortunately, are still categorically assigned. And they are assigned on the research that basically measure a survival based on the treatments that are done in clinical trials. So basically, the only biomarker for majority of cancers are Kaplan-Meier survival curves, and there the aggregate population statistic is applied on an individual diagnosed with cancer, and this is unfortunately how majority of cancers are treated still today. So basically, the survival upon the treatment is most probably the only biomarker that we have here. So once we apply the categorically assigned treatments, the outcomes can be in 4 different ways. So the drug can be toxic and not beneficial, which is the worst outcome. Then drug can be beneficial but toxic, and it's also not a favorable outcome, and not toxic and not beneficial. So there is only one group of the people diagnosed with the same cancer that will have no toxic effect and the benefits. And you can see that that is a really very small population of the people, but that is unfortunately the only way how the treatment has been assigned. So ideal thing is if we have a diagnostic biomarkers or any kind of biomarkers that can guide the treatment. And that is called also precision medicine or evidence-based medicine. In the essence, it is basically matching diagnosis and treatment strategy to each individual unique molecular makeup, whether this is genomic molecular makeup, proteomic molecular makeup, or metabolomic. Ideally, it should be based on systemic use of information about the patient molecular genotype, phenotype, and metabolic profile to tailor that treatment. Obviously, we are aiming to it. We are far away from it. One thing that would be ideal kind of scenario, if we take N of 1 as a single patient diagnosed with, to match this diagnosis with its unique molecular makeup. An ideal situation would be if our biomarker will be a driver of the pathological process, and it's at the same time as a drug target. So in that case, the only cancer nowadays, fortunately or unfortunately, is really the breast cancer, that we have 3 different molecular phenotype. So once we know that the cancer is HER2 positive, we can treat it with Herceptin, or if it is ER positive, with the estrogen receptor blockade, or the triple negative cancer, which are unfortunately most difficult to treat. But basically, if we can follow this scenario for any other cancers, we would be much better off, and the patients definitely would be much better off in that respect. And in that case, we would like to look to shift of the paradigm, and this is basically now we have shift from organ-based to pathway-based oncology, because we do know that estrogen receptor pathway and HER2 pathway are different pathways and deserve a different treatment. So a little bit about proteomics. I would like just to say about what is the central dogma of biology. We have a DNA, which is transcribed to messenger RNA, and we have the protein. So all of those polymers or DNA stretches and mRNA stretches and polypeptides can be used to track the cancer, but we believe that the proteins as direct bioeffectors are more relevant to a given disease phenotype than the genes or mRNA. Although I don't want to deny that it's important to know the genomic phenotype genes, but still proteins are at the site of pathology, at the site of the tumor. And the other paradigm is there that we would like to use cell lines, animal model, and human data to translate this newest discovery into the new treatment, because there is a feeling that there is really a long gap between some basic discoveries that are applied. So translational cancer research is really a mode of research that we can speed up the discoveries made in the lab and apply them directly to the bench, or from the bench to the patient's bed, and get that process speed. The other thing also, what we can target as a biomarker, most of the targets nowadays, like Herceptin and chimeric antibodies, are really hitting the cell surface. Anyhow, 70% of drugs used in medicine nowadays are really targeting the molecular cell surface. So what I would like to emphasize that our lab has pioneered the methods to analyze cell surface protein and cancer cell line, and we are doing it for more than a decade. As an effort of that, we have developed a shotgun proteomic approach to look into the membrane proteins of the cell lines or even human tissue, using strong cation exchange as a second mode of separation of the peptides to really beat or increase dynamic range of analysis, and using also high-end mass spectrometry with high resolution and high mass measurement accuracy. So one of the studies that we have done early in 2006 is basically we are looking into the plasma membrane proteins of hypoxia-adapted malignant melanoma cell line. Proteomics is not trivial. We don't have a luxury to amplify gene products like you can amplify genes. So the sample preparation is extremely important part of the proteomic analysis. So here is the westerns that are showing that you need to use a couple of molecules that that are good biomarker of cytosol like actin, PCNA of nucleus, and transferrin receptor as a membrane. So what this slide shows basically that our plasma membrane preparation is devoid of actin, and the PCNA is diminished, and there is an enrichment of transferrin. So basically all those membrane preps is basically enrichment, and not that something that we can say that we have pure isolation of an organelle, but enrichment definitely can help us to make enrichment and conquer dynamic range. So this particular study we use also 18 or 16 labeling to do the relative quantitation of changes in cell lines which are exposed to hypoxia, and we were able to identify about 5,000 proteins there. Of those, about 700 are upregulated and 800 downregulated, and 3,000 proteins show no change in our study. So this is a mass spectrum for the people who understand mass spectrometry. Here is a base peak chromatogram. Here is the parent ion of light label and heavy label peptide, and we can see the MSMS of all of them. This is a transferrin receptor. It was the biomarker that we are looking as a biomarker of the purity of the plasma membrane prep, and obviously there are no changes, significant changes, and we do not expect them, but this is just educational to show that we can see heavy and light label peptide in control and experimental cell line. The outcome of this analysis, obviously all proteomic data should be always validated using orthogonal method. In this time, this is a fact, and we have shown subset of proteins to be dysregulated in hypoxia-adapted cell line, and we can see the upregulation of hypoxia-induced factor, glutamate transporters, and CD13, CD44. So there are the proteins that have changed, and you can see TFR was not shown to be affected by the hypoxia in these cell lines. And further, so those are mouse cell lines. We wanted to see how many of those findings which we found in mouse cell line applied and amplified in human melanoma cell lines, and we found that CD13 and SDF1 are only those which are found upregulated in human melanoma cell line, and we have also do qPCR and FACS analysis to prove upregulation or dysregulation of this protein. So that is one example how in our lab we conquer dynamic range and have better reproducibility once you isolate the organelle of interest. Another interesting study was done on the lipid rafts, which are the insoluble parts of plasma membrane, and in 2005 we get into the collaboration with Bradley Stiles, and they are looking actually what are the binding partners for iota B toxin in viro cells. So after we isolated viro cells, after we isolated the lipid rafts from the cells that have been treated with iota B toxin and the other cells which are controlled, we again use O18 or 16 labeling to show the differences. And basically after that analysis, we have shown that a couple of molecules that are showing upregulation more than threefold, and CD44 was the one that we at that time thought might be the protein that facilitates iota B internalization into the cells. So our collaborators takes the long run, and just 2 years ago we have published another paper, and they have used a lot of molecular biological experiments, and they have shown that indeed the CD144 promotes intoxication. So that is kind of studies that we would like to have, that you do discoveries using global proteomics for biomarker purposes or any other biologically relevant process, and that you can prove that and using classical molecular biology. So now our latest approach in looking into the cell surface proteins is basically we have a big initiative by NCI looking into the RAS-driven cancers. And why? Because RAS is an oncogene that causes the most difficult cancer to treat, and we would like to see if there is anything that is specific to the cell surface protein on the KRAS expressing cells, and then see whether we can use those targets as mostly the immunotreatment. So that is antibody-mediated attack can be developed in different ways. It can be used using monoclonal antibodies, which is used like in breast cancer. We would like to have an option to find other targets for other cancers. Then we can use also antibodies that can be used to conjugate and carry the like other drug like in brentuximab, in Vendatin, or just use nanoparticles coated with doruxibitine and can able to treat the cancer cells. So there are many options that we can use if we find the proper target uniquely expressed in KRAS related cancers. So just for the beginning, I just would like to show you the MCF10A cell line when it was transfected with the KRAS, how different it looks like, and we are going into this direction and trying to see what are the proteins. But you can see that even just using scanning electron micrograph, that you can see how different changes, how cells transform when KRAS is overexpressed or transduced. In this setting, we have used two methods basically. We use hydrazide targeted method, hydrazide based method, in-house developed, and then we use our method that we previously showed using strong cation exchange as a global method using shotgun proteomics. So combination of those methods, we expect to give us the best profile of the cell surface of the KRAS cells. So now the next step will be also what we have done before using animal models. So my presentation until now was focused on the cell surface then of the cell cancer cell line for the biomarker research or drug, but mouse models are increasingly used in looking into the biomarkers. So what we have done, we have published a study looking into the HRAS-driven hypertrophic cardiomyopathy in transgenic mice, and as you can see that morphologically, once the HRAS is activated by tetracycline, the heart is big, and this is normal heart, and histology and microhistology look at. So we would like to see what is the molecular signature, what is really happening in the mouse heart when HRAS is overexpressed. So we have used spectral counting and as intensity value for the cells. We have normal heart and hypertrophic heart, and we see here the mostly dysregulated genes. So that is typical proteomic workflows. However, we wanted to go deeper and to see what is really happening in that myocard at the molecular level. First of all, we wanted to see for the people who are really mass spectrometers, that we are able to identify HRAS in a mouse heart, and also that expression of the HRAS is confirmed with immunohistochemistry. That was something that we would like to show before we may think of relevance of our findings. So to look at the genes that are differentially expressed, we use ingenuity pathway analysis to see we are really hitting the RAS pathway. And what is important here, we have really didn't use any shotgun proteomics in this. This is only 8 micron thick slides, which are 3 slides per sample that were used because we wanted to use it in the frame which pathologists typically use. So we would just use the slides of tissue and analyze them with the mass spec. And obviously we have also confirmed that RAS pathway is upregulated in mouse, which are p-ERK and phosphorylated ERK is upregulated in that setting. What is also interesting, we have found that embryonal myosin is activated with RAS, which shouldn't be found in a normal heart. Once any mammalian is born, the heart goes out of the cycle, so heart cells cannot multiply anymore. So the enlargement of the heart goes only on the size of the cell. There is no multiplication there. And that we can see that is driven by myosin alpha and beta, which is actually embryonic myosin that shouldn't be found here. So basically that gave us much confidence that we are really looking into something that is driven by RAS, HRAS. Then we use the spectral counting and calculated the ratio, and this diagram shows that Wnt pathway in general is activated secondary to the RAS pathway activation, and our collaborators confirmed that the beta-catenin is really increased in the mouse heart. It is in agreement with our mass spectrometric data. So the one thing which is interesting with this study, basically what we have seen in this research, that constitutive ACE activation activates Wnt, and then you have the differentiation actually EMT. One thing which is interesting that we found another application of, or kind of confirmation of our findings, that if someone would like to make pluripotent stem cells and generate cardiomyocytes that can be used as a treatment for the myocardial infarction and to using stem cells, and we found out that the laboratory that is trying to determine the destiny of the pluripotent stem cells toward cardiomyocytes, the Wnt pathway has to be inhibited. So this is finding which has not really related, which is independent finding, which really confirmed that what we have seen, the differentiation is driven by secondary Wnt activation. And if you would like to dedifferentiate the stem cells to different to cardiomyocytes, we can see that VNT is, and here are the publications confirming that finding. So we are pretty happy about it, finding that independently that Wnt activation with pathway is really cross-talking to the HS pathway, at least in these transgenic mice. So now I would like to switch the gear and talk about translational research. We are lost in translation, so why? So I would say this, we understand very well cancer cell biology, and we were able to treat very successfully cancer in Petri dish. Then we also understand that there is a lot of animal models, cancer animal models, and we also understand the cancer biology in animal models. Once we try to apply our knowledge in those two models to human, we've figured out, and scientists agree and physicians agree, that a lot of those findings are really not applicable to human. So this is why I say lost in translation. We need to figure out what the findings in these models can be applied. So why we would like, where I'm getting there, that we need definitely to look in the human samples using genomic and proteomics, because data obtained from cancer lines or animal models are not always predictive of what is actually happening in human cancer in vivo. So basically, what I'm proposing, that is a typical pathway where we had biomedical research, basic research from bench to bedside using animal models and cell models and applied there. That worked very well, but definitely I think the other way, looking into the human samples, human tumor biopsies, and then apply them to the basic research, so that it was full circle. What I'm basically advocating, and which our lab is trying to do, to emphasize the other way, so that they are complementary. They are not exclude each other, but we should use the capabilities of having human tumors tissue and clinical sample and analyze them to make a full circle. So obviously, in that, saying that, there are a lot of challenges. So despite the recent advances of MS instrumentation, the translation of proteomics findings, that was really difficult. This is mostly because of extensive dynamic range of protein concentration in tissue and blood, and even it is even worse in tissue. So that because tissues are also perfused by blood, so the dynamic range is a big trouble in proteomics, and addressing it is definitely an important part of proteomic research. Numerous studies have clearly demonstrated that current MS platforms still struggle to identify lower abundance cancer associated biomarker, particularly in peripheral blood. So we really think that that also the poor experimental design of early proof of principle studies is focused exclusively on plasma and serum didn't really give too much help to the young field of proteomics. So in our lab, what we try to do is use basically divide and conquer philosophy, look into the different kind of the tissue specimens and use different kind of separation strategy to minimize and really address the dynamic range of the issue of dynamic range, so that we can really look at a low level important regulatory low abundance proteins. The one thing is also which need to be addressed before you start using biomarkers, like I said before, nothing can match easy way to get. Someone will give you easy blood sample to analyze, but do we really need to go to only there? But here I have listed the differences in what sample you would use. So if you use peripheral fluid, plasma or serum or blood, the biomarker is there is in low concentration because this is 4 liters dilution. The biomarker is outside of cell environment. We don't know where it's actually coming from and what has happened to this biomarker. But there is always the ease of getting these samples which people, the scientists would like, and to look only in plasma and serum. Then from the other side, if you look at the tumor tissue and the tumor itself, if we analyze by proteomics, the biomarker will be in higher local concentration. It can be investigated within the cellular environment. We can compare adjacent normal tissue to tumor. You can do some mechanistic studies, but it is difficult to obtain. And basically in this editorial we wrote, we actually wanted to say, no, you cannot say one or another. We really would like to analyze in parallel both tissues and body fluids. So once you decide to do the tissue, there is also the question which I would like to address. There is a kind of still the jury is out. The jury is out for whether you use fresh frozen or formalin fixed tissue. Again, formalin fixed tissue is good available. You have a lot of repositories. You don't need refrigeration for that. They are easy to store. But then the trouble is because this is pre-analytical bias there, because those are formalin fixed, difficult to extract the protein, immunoreactivity is lowered, quality of DNA and RNA is lower, and you lose basically metabolite. And it really is. So what our goal, what our take is, and we have published that we would like to look at fresh frozen tissues. They are difficult to store, but you can use proteomic, genomic, metabolomics at the same time. You have good immunoreactivity, unbiased quantitation. Obviously, you know, availability is not great, expensive to store, and dynamic range issue is, but still we do believe that really if someone would like to look to biomarkers and drug targets, should use fresh frozen tissue. So the one thing to address it, we do know that removal of most abundant proteins in plasma and serum is well accepted, but typically, very frequently, it's overlooked that if this is a sketch of the tissue really has capillaries, veins, arterioles, lymph, and definitely the blood is the same problem that you have in blood, you have also in the tissue. So we really are the one, I think we might be the first one who try to deplete the tissue samples to facilitate that. And we basically have done tissue samples, normal tissue, tumorous tissue, and you can see after depletion how much of the albumin is there. It is, and this is the blood from the same patient. So basically, what we advocate that if you really would like to analyze at the same time blood and tissues, you most probably need to develop to deplete the to apply immunodepletion the same ways to remove albumin and most abundant protein from tissue samples, and we did it. And in that way, the most important thing is that we have developed an experimental design, a strategy that we should look into the parallel into the tissues and blood samples from the same patient. Blood should be developed, should be taken before the surgery, so that we can really have a fair ability to equally analyze at the same time blood, peripheral blood and tumorous tissue at the same time from the same patient. And we have applied this approach, we tested that approach on the renal cell carcinoma, and we can see, you know, we are repeating this paradigm. Right kidney and left kidney are the same genome, but you can see development of renal cell carcinoma clear type in that kidney. So those proteomes are different. So what we have, why we use the renal cell carcinoma, basically because this tissue is available, and still there is a high mortality of that cancer. 12,000 people have died in 2000 7, and there's no clinical biomarkers for renal cell carcinoma. So those are the reasons why we use this one. We could use any other tissue and to try to do the proof of principle study. So what our idea is to take a normal tissue, depleted, to take tumorous tissue, depleted, do the strong cation exchange, analyze it, and the idea is after that we localize the tumor proteins. Can we see them in the blood of the patient diagnosed with cancer? And that blood was collected before the surgery, so because after surgery there can be a contamination. We published a study in 2010, and now I'm giving another slide to be more precise about experimental design. So we are looking into the tissue, we're looking into the peripheral blood. Then we do the subtractive proteomics, and we look only into the proteins that are found identified only in a tumor, and those are the bona fide tumor proteins which are most probably biologically relevant. We didn't look at that first instance, which are the more proteins which are more expressed in tumorous tissue, but we thought that those which are found by mass spec only in a tumor might be biologically the most relevant one. And then when we do the subtractive proteomics between tumor and plasma, any protein that have been identified in plasma may be used as a biomarker. Now the question is how we do know that is something that is really coming from tumor, and that's a really simple idea that those proteins which are showing higher spectral count in tumor than in plasma, they are most probably German to the tumor, they are coming from the tumor, and those which are having higher spectral count in plasma are most probably not related to tumor. It could be some immune response or any other response to the tumor. So here is again our experimental design. We have used tumorous tissue depleted with the affinity depletion, normal tissue, and then we use preoperative plasma. Everything was strong cation exchange, fractionated and e-fraction analyzed in duplicate, and then that was subtracted to the bioinformatic meta-analysis. So basically what has happened in our analysis, we have seen about 1,000 proteins that are common between tumor and normal tissue, but there are 200 proteins that are found only in tumor. Then we look into the preoperative blood. This is a mistake. We have looked at preoperative blood, and then we found that 32 proteins are basically overlapped, but only 8 proteins showed higher spectral count in tumor, and this is the list of proteins that we were able to identify as a tumor protein in peripheral blood. You can see most of those are membrane proteins. Some of them are notorious, are not most probably renal carcinoma specific like pyruvate kinase, but some of them definitely should be considered as a tumor proteins that can be identified by mass spec. So that was our proof of principle study. And first we wanted to look at the cadherin 5, because most of the peptides that we identify from the tumor and also in the blood were really coming from the extracellular portion of cadherin, that we believe that might be the shedding of that extracellular portion into the blood. And we have found the reference that it is most probably coming from the endothelium of the tumor, because that is a protein that has been found and linked to the endothelium. Now when we talk about spectral count, so this is all total spectral peptides that we have seen from this protein in tumor, and this is 2 we saw only in a plasma. So this is the reason why we believe that this protein is coming from tumor, and we were able to identify that in a peripheral blood. Here is the mass specs, MS2 spectra of this peptide, and someone who see this, you can see this is in a tumor. There's very little of background. I don't think that you can see here intensity, but it's 2 orders of magnitude that this peptide is less abundant in plasma, but still one can see that this is the same peptide that we see in patient's tumor and the plasma. We have done extracted ion chromatogram also showing that the much lower abundance of this protein in peripheral blood. So that was a mass spectrometry take on it, but no one obviously nowadays would really consider mass spec solely as a bona fide identification, and we have used the Western to look into cadherin 5. We look in patient serum. We look into the serum of the patient using 2 different loading amount, and we use HUVEC cell line as a positive control because this protein is there, and then we use a cell line of prostate and ovarian cancer, and we really don't see that. So we believe that that definitely is a good indicator showing that that protein may have an important role in endothelium of the tumor. And we have taken a peripheral blood of 4 patients, and we did serum, Western blot of serum for, this is our initial patient, other 4 patients, and out of those 8 proteins that we have shown before, 4 we were able to prove that they are found in a patient with diagnosed with renal cell carcinoma. Those sera we get from the Dr. Linehan from NIH. This basically showed that the value of cadherin 511, DDX23, and pyruvate kinase as a panel of the biomarkers for the renal cell carcinoma, at least for this patient, another 4. Obviously, to extend it to the to real biomarkers, there should be a lot of clinical studies and a lot of samples to be run. So here I would like to mention, most probably in discovery phase, we don't need a lot of samples to analyze, but once you narrow down the panel of biomarkers, they can be tested on the thousands of patients, and you don't need to use mass spec. You can use ELISA or any other or immunoassay to see in how many patients these biomarkers are really show as a positive. We look into the pathway analysis, and we look what could be the cadherin 5 potential interactors to satisfy our curiosity in cancer biology, and protein of interest are definitely Zagata, Wnt, tumor necrosis factor, and at least we have shown that that protein has some important cancer biology interactors. So that is something that we would say it can be used cadherin 5 the same as a HER2 because it's a biomarker driver, and most probably is a driver in angiogenesis rather than in the epithelial part of the cancer. So after this analysis, we actually found a study that was a genomic study, and we really wanted to see it's a genomic study of the same tumor, it's clear renal cell carcinoma, on much bigger cohort of the patients, and we wanted to see how our analysis coincide with the findings of genomic data. Actually, this slide shows just how much of those proteins are relevant based on other studies. So basically, what we wanted to show here that there is what is happening. All these cadherins that we believe that is basically mesenchymal cadherins are shown in the tumor, and they are most probably the part of the TGF-beta driven epithelial to mesenchymal transitions. And this is again the slide which shows how many of cadherins we are here, and that we really do believe that in renal cell carcinoma biology, that cadherin switching is a phenomenon that is definitely drive that. But it was really hypothetical, and we wanted to check that in the and then also what we wanted to see that another gene which we looked later on in our data which is overlapping, which is PLIN1 or adipofilin that we found that is 9 times more increased in tumor, and then we ran the Western with both really the tumor lysis of renal cell carcinoma and show significant upregulation of that protein and fatty acid binding protein in renal cell carcinoma. We also found out that adipofilin is used in a vaccine of renal cell carcinoma, and that is a part of something which is recognized as activation of lipogenic pathway in a variety of human cancer, and in renal cell carcinoma it's a mostly activated pathway. So this is the paper that I wanted to show that we have been comparing our data to. It is basically the part of genomic analysis. So the Chun et al., they have used 10 non-metastatic early-stage renal cell carcinoma, like was our in our. And then they use patient-matched normal adjacent tissue. So we have N of 1, they have N of 10, and to see what really happened there. So they have used Affymetrix, and they found that there is 33,000 well substantiated human genes that can be followed in these tissues. And based on ANOVA and p-value less than 0.05, they found 13,000 genes that can be considered as significantly changed. Obviously, we cannot, we don't have a luxury to amplify proteins, but anyhow, their results were that the unique pathways that are really depicting renal cell carcinoma is basically the features are carbohydrate metabolism dysregulation, loss of normal renal cell function due to the epithelial dedifferentiation or EMT, and this adipogenic trans differentiation. So this group, they have done cross-validation themselves. They have used qPCR and immunohistochemistry to confirm the initial findings. So we wanted just to see how that looks in comparison of on our data. So the first thing that they have looked in the qPCR of their data, that they have found the upregulation of genes involved in carbohydrate metabolism, and that really coincides with our data, and the relative concentration of protein expression via spectral counting also revealed changes in carbohydrate mechanism, similar to this observed by Chun et al. We also saw the different from their study upregulation of gamma amylase 2 and adenomedullin in parallel with downregulation of fructose biphosphate aldolase. And these findings really can be something that we can say that our data goes well with the genomic data, and proteins that are highlighted here being identified in the proteomics. Obviously, a lot of gene products we didn't see just because of the dynamic range. But anyhow, we can say that we also see the dysregulation of carbohydrate metabolism in renal cell carcinoma. So there's this is the proteins that we have been identified that are highlighted in red, and this is the immunohistochemistry that Chun et al. Performed for analase 2 and aquaporin 2. So these findings allow us to hypothesize that the switch from isoenzyme B prevalent in normal kidney to isoenzyme C predominant in renal cell carcinoma is consistent with simultaneous glycolysis pathway activation, and this is Warburg effect, and this is something that we can observe together using proteomics and genomics. The next thing is also that we have been talking before, and that confirmed, the genomic analysis confirmed that EMT is a major process driving renal cell carcinoma tumorigenesis. Again, we have also seen vimentin as upregulated in renal cell carcinoma and in cadherin as well. So that gives us another confidence that our approach is something that definitely can be followed further with genomics, and we really do believe that proteogenomics analysis studies will definitely will be important. So what we are showing here, really, the proteins that we found that are in sync with findings of genomic study. The proteins that we found differently expressed are not really the part of this presentation. We use this just to show the proof of principle that our experimental design gives data which can with high relevance to the pathology under the study. And also this lipogenic pathways activation, this group also did immunohistochemistry on adipophilin, as we have done using our Western it is again, and there is also other proteins which are fatty acid binding proteins, different isoform, differently expressed in renal cell carcinoma versus normal tissue. So to recapitulate this, I just wanted to show that combined tissue blood analysis in cancer biomarker discovery phase is extremely important. It is extremely important to look at the same time in a tumor and normal tissue and see whether the proteins that are tumor proteins can be identified in peripheral blood, use as a panel of biomarkers, use pathway analysis and spectral counting as indication of intensity, and to get data which are meaningful and can really help out to understand biology and elucidate biomarker and potential drug targets. So what is the translational aspect of our research? So the clinical proteomics, the way we do see, which is really molecular profiling of solid tumor on the individual basis, using tumors and blood together, can help two ways. It has two ramifications, basically three ramifications. It can help a biomarker discovery, a panel biomarker discovery as shown on our principle experiment. Can also help doing mechanistic studies and also form a cancer proteome atlas. So obviously every tumor heterogeneity, I didn't mention that, is a big issue. So every tumor, although we classify it as, for example, renal cell carcinoma, it's an individual signature. So the more tumor tissue samples and blood samples are analyzed the way we propose, the more information we can see and not battling then N of one, but N of many to get closer to the novel biomarkers and drug targets. And we see the proteomics as a complementary part of the omics, although I think we can see the light in the end of the tunnel that proteomics may be increasingly more successful and a better tool to look at the cancer biology and drug targets and novel biomarkers. So what I would like to conclude here. So we have developed mass spectrometry-based clinical proteomic pipeline and demonstrated its capability to detect tumors protein in peripheral blood in our proof of the principle study. We really believe that if you look in parallel in tissue and peripheral blood specimen on individual basis, it's critical for mass spec-based biomarker research that would allow better characterization of tumor phenotype and pathways implicated in pathology under study. We do believe that depletion of abundant blood-derived proteins from tissue specimens should be routinely used in a mass spec clinical workout, and along with the depletion of plasma and serum, and we think this is really critical. And to increase dynamic range and protein coverage, it is important to use multiple dimensions of separation using shotgun proteomics with high resolution and high mass measurement accuracy. Without state-of-the-art mass spec instrumentation, it will be very difficult to look into the complex human clinical specimen. And this technology that we developed can be used along with genomics and metabolomics for integrated analysis of clinical specimen in the context of cancer biomarker discovery and drug discovery. The results from our proof of principle study, we have published in 2010, as well as the result of similar succeeding investigations, suggest that small number of samples may be sufficient in the discovery phase, providing that initial finding can be cross-validating using orthogonal immunoassays. And I would like to acknowledge our collaborators, Marcel Lillingham from NIH and Deborah Sitchin and Be Very Wrong, and Bradley Stiles from Yosemite and Duke Stocking from Leidos. Our director of Cancer Research Technology program is Dwight Nisley, and we are now involved in the RAS project. Gordon Whiteley and is a lead on the cell surface proteomics, and this is showing who did a lot of proteomic data. Daru is our expert of tissue depletion, and Ting Chan is the guy who is an extinguished chromatographer who is responsible for our strong cation change, and Brian Luke does our statistical analysis. So that would be all, and I'm now open to questions. So then I'm finished with the presentation, and we can see how we can move forward. Hello?